Well, I'm a food engineer, so I will have a slightly different perspective from the previous two speakers who really laid out a great vision on the field. My vision will be more technologically oriented. In general, I hope that the challenges that we see are similar in the sense that we do see, I think, challenges around environmental impacts, around achieving zero hunger, a resilient production all over the world, and of course also public health, which is very closely related to nutrition, obviously. Well, my primary focus will be on environmental impact, but I will hope to show you that it does have its relevance for the other aspects as well. So basically what we see is that there is a great need for the production of more protein, more high quality protein, to sustain the people in the world. And that's the reason why we have seen the big transition, the big protein transition, for example, towards meat replacers. And that's obviously fueled by the fact that the production of plant-based protein requires much less land, in this case expressed in the form of equivalent CO2 emissions compared to different types of meat. However, this is only part of the picture, because the proteins in plants typically have to be processed before they can actually be applied in high quality foods. And if you take that into account, the picture changes slightly. Processing is quite important in terms of equivalent CO2 emissions. It really imposes a significant footprint, which is mostly related to the extraction of the proteins from the plants into high quality food ingredients. And what we typically see is that in the production of a protein isolate, the process actually only takes part of the proteins out of the matrix. What we typically see is that a legume is of course first milled. It is then suspended in water. There's usually some separation of starch or oil, and then the proteins are dissolved at, for example, elevated pH. They're then precipitated at their isoelectric point, it's neutralized and dried. The drying takes a lot of energy, while the precipitation process does not precipitate all proteins. In fact, not even all proteins are dissolved as well, which means that only part of the proteins end up in the protein isolate. Well, that means that, in fact, for the production of 1 kilogram of protein, we actually need more raw material than we would need if we would have a more efficient process. And that really has impact on the total efficiency of the way that at this moment we create these plant protein ingredients. Well, things can also be done differently, and that's the subject of an emerging technology, which is dry processing. So not use this dissolution precipitation process, but actually using a mechanical process where a typical legume, for example, or other crop is milled, and then the different particles that are created by this milling are separated, for example, by air classification. The typical protein bodies in the seeds are much smaller than the starch granules, and that means that by simply blowing at it, you can actually easily separate them. The big advantage is that you do not dissolve the proteins, which also means you do not need to recover them again by precipitation, and therefore, in principle, you can make use of a larger part of the proteins in the resource. Another advantage is the fact that you actually do not process the proteins at all, so they're not just in their native state, but they're even in their native protein bodies as well. So the natural structure, in fact, is retained. The disadvantage of this method is that the protein you get is not pure. It might be up to 70% of protein, between 50 and 75% of protein, which means that the properties of such a ingredient will be really different from the high purity protein isolate that you produce via the conventional route. You can improve that by, for example, adding a second step, which could be, for example, also a dry step in the form of electrostatic separation. This has been a traditional way of working actually in a number of other fields. For example, in the ore industry, the mining industry, electrostatic separation has been applied on huge scales. So by applying this technology and transferring it towards the food area, you can actually achieve much better separation. In this case, you basically rub the particles along a surface, which then gives them a charge, and the charge depends on the material of the particle. Just like this small boy on the lower right corner, if he rubs his trousers over the slide, he will also get a charge which depends on the material of his trousers. Just like, and that's the same with proteins and carbohydrates, for example. If you first give them this triboelectric charge and then separate it via an electrostatic field, you can actually increase the purity of the proteins. It's still not as high as with the protein isolate, but at least a bit higher. You can improve it even further by going towards something like hybrid gentle fractionation, where you start with a fraction, and then the particular fraction that you're interested in is not subjected to a very rough wet treatment, but you basically just suspend it in water and then use sedimentation to separate the different parts. Well, by doing this, you get fractions which are almost as pure as the conventional ones, but still the proteins are very much in their native state. They still retain their proteasome structure, and they do have quite interesting properties. What we typically see is that the yield is a little bit lower. So the typical yield you get with dry fractionation is really best, and with conventional it is actually significantly lower. But with this hybrid technology of first dry and then wet separation, well, also the yield is a bit lower, but we see that the purity of the proteins is in fact similar to the conventional purity obtained with conventional wet separation. Still, you do not use that much water, and that's simply because you only take a small part of the total crop and you do not put everything in water, which actually makes a big difference also in the processing of the other fractions. And quite related to that is the fact that the energy use is then subsequently also a lot lower, simply because a lot less water has to be evaporated. So here you see sort of the best of both worlds. You do compromise a bit on the yields, but actually you go towards the better purity of the proteins, but still retain much of the benefits in terms of water use and energy use. So that could be a sort of compromise, which would really help us make the whole processing more sustainable. But that actually might not even be necessary, because if we look at the typical rough ingredients that we can get with it, which maybe 50 to 70% of protein or so. Similarly, for starch, we can have something like 70% starch and 30% fiber type materials. We see that these materials actually have quite interesting properties, and we might be able to use them directly in nice foods with a number of advantages. For example, the fact that they will contain more fiber might also have its impacts on the nutritional value of these components. But the point is, the functionality of these ingredients is in fact a bit different. Well, if I start with the first one, the starch, what we typically see is that by simple dry fractionation, you can have a starch concentrate, if you want, of about 68 to 70% of starch, which leaves about 30% of fiber in this fraction. Well, surprisingly, the viscosity that you can reach with this starch is quite similar to what you can get with pure starch, which is produced in a conventional way. In the upper right corner, you can actually see that the commercial pea starch has almost the same viscosity at the same level of solids. So in fact, a higher amount of starch compared to this what we call functional starch fraction. In terms of syneresis of the gel that you can get, in fact, you see slightly better behavior, but that might not be completely significant. The main point is that we see similar properties, while at the same time we still preserve about 30% of fiber in these starch gels, which I think might be significant from a nutritional point of view. And in fact, also significant in terms of the use of the raw materials that we have. If we go to the proteins, we see that these functional fractions can actually be pretty good emulsifiers. For example, what we see is that if you use these proteins as emulsifier and then bring the emulsion to the isoelectric point, the emulsion remains stable. And even if you have freeze and thaw the emulsions for a number of times, you see that they all remain stable. While if you do the same thing with the commercial, more pure protein isolate, you see that the emulsion becomes unstable. In general, if you look at plant proteins, like, for example, pea proteins, we see that commercial pea proteins are actually relatively poor emulsifiers. But if you take these functional fractions, they can be much better emulsifiers. Typically, what we see is that that also depends on the composition. The commercial protein isolates, in this case, once more exemplified with P, mostly contain globulins, because the albumins are solubilized, but are not precipitated, because they typically are much better soluble in water. So we typically normally lose the albumins in the plants, in the wastewater, basically. But if we take these dry routes, or the more gentle aqueous routes as well, we can preserve the albumins. And these albumins are actually excellent foamers. They're similar to whey protein isolates in their foaming capacity, and that's definitely not the case with the globulins. Conversely, the globulins are actually more suitable as emulsifiers, while the albumins give flocculation. And by simply steering the way that we process these plants, we can actually have more albumins or more globulins, or actually recover them both, but having them in different fractions. And what we see is that with the albumins, the typical overrun that we can achieve is very high, while the half-life of the foam is also quite high. So you can really see that that these albumins, if we actually take the efforts to preserve them, can have very high quality. And well, just remember that this was a fraction that we typically typically lose in conventional processing. And well, actually, we also see that these albumins are important in creating gels. One of the big issues with, for example, pea protein is that they're not very good gel formers, which has its consequences, for example, in their use in meat replacers. So what we typically see is that if we do have these albumins, we can achieve much stronger gels, which really makes them much more suitable for a number of applications. If we compare in the graph the gel strength that you can obtain with commercial pea protein compared to the albumin retaining gel, then we see 2 orders of magnitude of difference in gel strength. And of course, that opens up possibilities. So that really shows that we can actually have pretty good functionality of these fractions, even when we actually have much better use of the raw material and much lower use of energy and water in the production. And it's the same with oils as well. Of course, commercially, oils are produced by expressing and then also using hexane, for example, to extract the oil out of the raw material. But you can also do it differently by simply using water. If you take the seeds, for example, sunflower seeds or even soybeans, you blend them in water. This can be done in the kitchen machine, but can also be done on a much larger scale. Then filter out the solids. You actually get an emulsion which still contains the original oleosomes, which still retain their phospholipids and the typical proteins which actually protect the oil against oxidation. So that means you get an emulsion which you can apply directly in a food, which does not need to be protected anymore against oxidation, because we preserve the natural mechanism for this that was already present in the seed. In fact, you can do a lot of nice things with it, like, for example, spray drying. They're quite stable, and you can then redisperse them in water if you want to apply them in the product. So also here we see that we can actually really lower the footprint and actually have much cleaner production by avoiding the use of chemicals, like, for example, hexane. We can reduce the amount of waste by retaining more of the valuable material in the various fractions that we get. Well, I'm not a nutritionist, so this is just speculation on my side. But I think that in terms of nutritional value, this less refining can retain more fiber and other components, which might actually help in the total nutritional value. There are a number of routes to mitigate or remove anti-nutritional factors, but that's an area where we really need some more research. But the interesting thing is we can preserve some natural functionality like antioxidation, for example. And that might also be attractive for consumers. If we want to have product with less additives, then it might just be a nice idea to retain the functionality of some of the raw materials which was present in the seed, and we now simply retain in this ingredient by retaining part of its structure. Now, I was also asked to tell a bit about costs, and in this case, we actually go towards less processing, which means less capital expenditure. So in principle, in terms of hardware costs, I think the intrinsic costs can be quite a lot lower. However, there is this issue about the functionality, which can be quite high, but it's also complex because we have very complex ingredients, which also means that their behavior and their interactions can be much more complex. So the positive thing is we can actually combine improvements in sustainability, nutritional value, attractiveness, and also costs. The disadvantage is that we need to invest a lot in understanding these materials. So therefore, these functionalities are really fundamentally different from refined ingredients. The different components will actually contribute to it, but we still need to find out exactly how this works and how we can predict it. So this complex functionality has to be captured and has then to be used in the formulation of complete food products out of these ingredients, which will need significant research. Well, basically what we see is that this gentle processing actually suffices to achieve most formulations. For example, here a portfolio was chosen of a number of products, and well, the typical freedom of formulation that we have with relatively pure ingredients is not really necessary. If we have sufficient refining to more or less cover the window that we need, that should be enough. But then, of course, the challenge is to predict the functionality of these mild fractions such that we can not only have the composition, but also the properties of the product right. And we have set out to use data-driven methods here because the mechanistic way is difficult because of the complexity of these components. But with that, we hope that we can be able to capture the individual functionalities and then optimize using these data-driven models, not just on the functionality, but then also on for example, the global warming potential. And this is just an example where we did this for viscosity, so just one functionality. And what we typically see is that if we then have a numerical optimization, we can actually achieve exactly the same properties, in this case viscosity, by substituting the isolates, the pure commercial components, which are colored red here, with these mildly fractionated components, which are green. And then we see that we actually need less dry matter. We can achieve the same properties, and we can also substantially reduce the global warming potential. Obviously, this needs a lot of research, but it might show how we can actually move forward if we combine the production of the ingredients with the application of the ingredients. And that's, I think, an essential part. We basically have to realize that a food is not just a set of molecules, but the structure is not only important in the end product, but also in the ingredients. And if we then learn why the natural structures are there in the first place, and then use them in our product, I think we can actually achieve quite a bit in terms of sustainability, while at the same time also adding to the nutritional value of these components. And that's, I think, not just the case for plant-based components, but if we go further in the protein transition, we will also see other types of ingredients, for example, fermented food ingredients, and ultimately also things like cultivated meat and precision fermented dairy. But also there we see that the production of pure ingredients is difficult, costs a lot of energy, and also wastes quite a bit of the raw material. So I think it's even there, it is important to see what we can do with these impure ingredients, and then try to capture and understand the complexity and natural functionality of these ingredients. So that's basically what I would like to discuss with you. I would like to thank my whole group, but also the different partners that we have. I do think that this is something that's where we can actually combine a number of the challenges that we face towards a more sustainable future. Thank you very much.